Hi, my name is Sharia, and today we'll be talking about basic cell theory and microscopy, as well as some extra info. Starting with cell theory is a group of statements that together serve a general tenet for life on Earth, or a foundational understanding. The three main components of cell theory is that all living things are composed of cells, cells are the basic units of life, and all cells come from pre-existing cells. Starting with the first one, all living things are composed of cells. Now, some things that qualify as living things are bacteria, fungi, animals, and plants. However, viruses are not considered living because they need a host in order to replicate and reproduce. And since it can't reproduce on its own without a host, it's not considered alive. Now, everything that's alive is going to be composed of cells. There's nothing alive that isn't made of cells. If it's not made of cells, then it's not alive. Now, cells are the basic unit of life. Cells are the smallest singular unit of life that you can have. Now, the cells themselves also have tons of components inside, such as nuclei, endoplasmic reticulums, ribosomes, so on and so forth. And each of these components aren't considered alive, but the cell as a whole is. So, in this picture down here, you can see from going to an atom all the way up to a person. And around where you would draw the line for what's alive and what's not would be right along here. From atom, molecule, macromolecule, and organelle to cell. Because the cell is when it starts having all the components needed to perform the processes of life, such as homeostasis, reproduction, etc. Now, all cells come from pre-existing cells. On to Earth today, the only ways cell can reproduce is from other cells, through things like mitosis, meiosis, fission, etc. Except, the only exception to this would be in the original origin of life. Since if cells can only come from other cells, that creates a paradox where no cells can ever exist, since you'd never have cells to start the chain with. So, at the original origin of life, it is theorized that cells may have come into existence spontaneously. However, it is also theorized that that occurred due to the extreme conditions at the time of a prebiotic Earth. However, nothing is really known yet, and today it's very difficult to recreate those same conditions, so we can't just create life or create cells. Now, moving on to microscopy. Microscopy, however you say it. Microscopes are devices that are designed to produce a magnified image of objects that are too small to be seen by the human eye. Developments in microscopy have also led to discoveries in biology, significant discoveries in fact, such as just seeing cells, viruses, bacteria, as well as organelles, components of cells, and so on and so forth. Starting with magnification. Magnification is how much larger an object appears compared to its actual size. And that's what a microscope is all about. Making small things bigger so you can see them. Now, in order to determine the total magnifying power of a microscope, you need to, for a light microscope at least, you must look at two things. First, the ocular. Here, the ocular is going to have its own magnification power, since there are lenses in here, and the objective, as these are also going to have a lot of magnification power. To find the total magnification of your microscope, you need to look at the ocular and the objective and multiply those two things together. So total magnification is going to equal to the magnification of the ocular times the magnification of the objective. For example, if the magnifying power of the ocular is 10x and the magnifying power of the objective is also 10x, then the total magnification power would be 100x. Now looking at how to use a light microscope, as well as components. Starting with the components, first you have the eyepiece, the ocular lens. This is what you look through to see the specimen. Next is the body tube. This is what houses the lenses for the ocular, the eyepiece. The revolving nosepiece is what allows you to switch between objectives stably. So you can switch from 10x to 4x to 40x, and so on. The arm holds the objective and the ocular in place. The objectives are the magnifying lenses themselves, and you can switch between stronger and weaker objectives depending on what you're looking at and what kind of image you need to see. Now, stage clips. Stage clips here are what hold the slide with the specimen on in place to stop it from sliding around, so you can get a clear image. The stage is just what holds everything up. It's this black thing right here. The coarse adjustment knob raises and lowers the stage here to help with magnification. This here just keeps the stage like together. It's like a screw that just holds it all together. It also focuses the light from below here, which is what allows it to be a light microscope and helps it view the specimens by lighting up the specimen. And this here is just the base. The fine adjustment knob is what you can use to help really see what part of the specimen you want to look at and just focus in on it more specifically. So how would you use this? First, you would start by placing a slide on the stage so that it's centered up under the objective lens here. Then you would turn the revolving nose piece so that the lowest power objective lens is clicked into position. That would be this one, presumably here. And while looking at the objective lens and the stage from the side, you should turn the coarse focus knob here, so you're not looking through the eyepiece yet, so that it lines up with the While looking at the objective lens and the stage from the side, turn the coarse knob so that the stage moves upward towards the objectives, and move it as far as it will go without touching the slide. Look through the eyepiece and adjust the light source and the diaphragm until you can attain the maximum comfortable level of light. Then slowly turn the coarse adjustment so that the stage moves down, away from the slide, and continue until the image comes into broad focus, or you can kind of see something there. Then turn the fine adjustment knob as necessary for perfect focus. Then move the microscope slide on the stage. Sometimes some microscopes will have knobs you can turn to accurately move the stage so you're not just pushing it around. And move the microscope slide until the image you want is in the center of the field of view of the microscope. And then readjust the light source or the diaphragm in order to attain the clearest possible image. And once you have a clear image, you should be able to change it to a higher power objective lens with only a minimum use of the fine focus knob. And if you can't focus on the specimen, just do those steps again and work from objective to objective until the higher power objective lens is in place. Now, how about preparing a slide? Wet mounts are very important to preparing slides because it allows you to see parts of cells, especially through staining. In a wet mount, a drop of water is used to suspend the specimen between the slide and a cover slip. And the procedure is used in a laboratory to observe motile organisms like bacteria or samples that need to be stained prior to viewing with a microscope. Now, when it comes to staining, there's a couple different types of stains. There's iodine stains, which is one of the more commonly available stains and is used to identify starch in a variety of samples. And it will stain starch as brown or blue-black. Glycogen will show as red. Methylene blue is also useful in identifying cell nuclei and DNA. Gram stain divides bacteria into one of two types, gram-positive or gram-negative based on the size and composition of the cell wall. Gram-positive bacteria cells stain purple and gram-negative bacteria cells appear pink. Now talking about the cell or microscope field of view. The microscope's field of view is the diameter of area visible through the microscope. So if what you could see is inside this circle and you'll see a bunch of stuff here, this whole thing would be the diameter of your field of view. Now there's a couple different ways to find the diameter. If the microscope's on a low enough power objective, you could just place a transparent ruler underneath the microscope and just measure the field of diameter straight from that. However, on higher magnifications, that can be more difficult. So you can calculate that using an equation. One is starting out finding out the diameter using this method of a smaller field of view, then taking that diameter with D1 as the diameter of the smaller field of view, used from a lower power objective, and multiply that by the magnification of the low power objective. So you have the diameter at that low power objective and the power objective itself. Then you divide that by the magnification of the high one, or whatever higher power you're moving to. And this will give you the diameter of your higher power objective. Now, how about estimating the size of the specimen? Well, the first thing is to just estimate the fraction of the field of view that the specimen occupies. So if you have a big specimen that takes up like roughly half of the field of view, you can just multiply the FOV diameter by that estimated fraction. Another method is, if it's smaller, you can just estimate how many of them would fit across the thing. And you could just multiply whatever number you come up with the diameter from earlier, and that would give you a number as well. Now, what about drawing specimens? Because once you have information or a picture from your microscope, then you have to draw it, oftentimes, if you can't just take a picture. Now, drawing is a very important skill in biology and is considered a type of qualitative data collection. When you're drawing a cell structure seen with microscopes, drawing should be done in pencil on white, unlined paper, so that you don't have anything extra in the diagram, and if you make a mistake, you can fix it. The picture should probably be in the center of the drawing, so it gives yourself the most room to draw and to add labels later on. The drawing should be large relative to the page, and it should occupy at least half of the space. Labels should have straight horizontal lines, and they should form a vertical list. And all labels should be printed, not in cursive. When it comes to technique, lines should be clear, not smudged, which may happen when you rub your finger over a line right when you write it and the lead's still fresh. Also, the scale should be included to estimate the size of the sample with the correct number of digits and unit. Another thing is accuracy. You should really only try to draw what you see and not try to add in what you think should be there. and not necessarily draw every single thing in the field of view, just what's asked for or what's important, since drawing extra things is time-consuming and unnecessary. For example, when drawing on a lower power plane or objective, you shouldn't have to draw every single cell there. Now, this is an example of a drawing of a cell under a microscope here with the plant cell. Here, the student is drawing a single cell from this microscope that they got, or the results from a microscope they got. And they draw whatever it is they can see, these green chloroplast-looking things. Yep, chloroplasts. And in the cell walls, the cell membranes, the cytosol, so on and so forth. This is everything they could see, so this is everything they put. Nothing extra and nothing less. They also included the magnification, even though they could be more specific, as said in the annotation. Same with the title, they could be more specific. But other than that, the drawing is good. And it's probably better than anything I could draw. Now, moving on to discoveries, or I guess extra information when it comes to the field of microscopy. Microscopy. I'll get it eventually. Microscopy. The invention of the microscope in about 1600 and its improvements over around 400 years have resulted in significant advances in our understanding of the microscopic world, as well as extremely important advances in biology specifically. The microscope is really important in basic science, and it gives the observer a view of a world that we can't see on our own. When it comes to developments in microscopy, the most significant is one of the most significant would be the development of light and electron microscopes. Both of them are microscopes. You use them to view cells, but what is the key difference? Well, light microscopes have a much less powerful magnification compared to an electron microscope. So let's talk about that. The way a light microscope works is that they view living specimens using their natural colors, or in their natural colors. These microscopes usually use a glass lens to bend light in order to magnify images, and the extent of magnification is dependent on the lens used. The clarity of cellular substructures can be improved via the use of fluorescent labeling, synthetic dyes could be good for that, or immunofluorescence staining, which uses antibodies that are conjugated to fluorescent probes to specifically target a cellular component of choice, if you're trying to highlight specifically mitochondria or specifically ribosomes in certain cells. So in essence, the way a light microscope works is light moves through a condenser or lens, then goes to the specimen here on a slide, then goes through the objective on another lens, until it eventually reaches your eye. So that's the way a light microscope works. Now let's go to an electron microscope. Electron microscopes can generate images at a much higher magnification and resolution. However, they can't view living specimens in natural color. These microscopes use electromagnets to focus electrons and produce monochromatic images, which means that they create images with no color. It's always black and white. Now, you may add false color if you have an idea of what that color may be, but you'll never get an actual color from an electron microscope. The two main types of electron microscopes that allow for different visual representations of biological specimen are transmission and scanning electron microscopes, also known as TEMs and SEMs. TEMs pass electrons through a specimen to generate a cross-section image. So if you have a full cell, if you pretend this is 3D, it would only give you that sliver of the cell. Now, a scanning electron microscope, they scatter electrons over a surface to differentiate depth and map in 3D. So if you had a cell, it would send a bunch of electrons out all over the place, and it would basically just map the depth, create like a sort of map of the surface, like a topographic map. Now, cryogenic electron microscopy, which is it's a way of preparing samples. It involves freezing the sample prior to viewing in order to generate images of a comparable standard to X-ray crystallography. This cryogenic electron microscopy allows for the determination of molecular structures at near atomic resolution without requiring the crystallization of the specimen. If the frozen specimen is cracked along a plane via freeze fracturing, then internal cellular structures can be studied. So basically, by doing this, they would essentially just crack the cell in half, and they would get a cross-section of it that they could study. So the way an electron microscope works is you have an electron source instead of a light source, which move to a condenser, which then goes to the sample. Or no, not the sample. It goes to an objective to help further magnify it. Then it moves to the specimen, before then making it to a detector and then to your eye. So that is how an electron microscope works. In order to compare magnification, you could compare from an electron microscope to a light microscope, and you would get this. With this being the same molecule or cell, the resolution of the electron microscope is much greater than the resolution of the light microscope. And you can actually see these circular structures here, which you can kind of vaguely make out here in the light microscope. So that's another difference in resolution between light and electron microscopes. Anyways, technology begets discovery. If technology is machinery and equipment developed from the application of scientific knowledge, it lets us eventually, an advancement in technology leads to the discovery of something new or learning something new for the first time. So as technology advances, discoveries will advance as well. Anyways, as tech continues to advance, new microscopy techniques get developed. In fact, there's a newer one that's trying to combine light microscopy and electron microscopes in order to create detailed three-dimensional images of cells. So instead of just getting a 2D cutout of a cell, you have like a whole sphere, and you could see the different parts of it, and you could look at different cross sections or whatever you wanted to, or you could look at the whole thing at once, like a giant snow globe. Anyways, that is the basics of microscopy and cell theory, as well as a little bit of extra information into the history of Microscopy. Thank you.